Greetings. I'm Robert Lee Kilpatrick. I'm the chair of the Health and Medicine Member-Led Forum here at the Commonwealth Club and also a trustee of the Board of Governors. I'm really excited about this program today, and I want to thank all of you who are here in person as well as an online audience. Those of you who are here, please turn off your phone. The last time I said that, halfway through, my phone went off. And come on in and just have a seat. So I'd like to introduce briefly our speaker, whose biography was so heavy I couldn't carry it. So I've managed to have a succinct version of it. So the title is Giving Scientists Superpowers, How an AI-Powered Thinking Microscope is Fast-Tracking Medical Breakthroughs. And one of the things I discovered was, may I call you Steve? Steve, Professor Steve. He's the director of the Gladstone Center for Systems and Therapeutics. He's a senior investigator at the Gladstone Institutes. He's a director of the Hellman Family Foundation Alzheimer's Disease Research Program at the Gladstone Institutes. He's a director of the Taubie Coret Center for Neurodegenerative Disease Research at the Gladstone and a professor at the Department of Neurology at UCSF. So I think you're super qualified to give this talk today. So without further ado, I'd like to invite you to come up to the podium to use your slides. There are question cards on your seats. There are some pens floating around. If you have a question, please fill it out, and my assistant, Barbara, will collect them and give them to me, and then I will receive questions from the online audience too. So without further ado, Professor Steve.
Thank you. Thank you for such a kind introduction. Yeah. So excited to be here. I was in Princeton yesterday giving a talk on agentic AI and robotics, but flight home this morning. So looking forward to being with you tonight. Thank you for coming out. And I hope this will be a fun time for you and really encourage you to ask questions. I think it's a lot of fun for me, actually. So let me begin by just telling you a little bit about this place you heard of, the Gladstone Institutes. It's an independent biomedical research institute located here in Mission Bay, close to the Warrior Stadium and the Giant Stadium. And it was founded in 1979, and it's continued to grow over the years. We now have a $125 million operating budget. We have over 600 scientists that are divided into 35 laboratories, including 2 who've won the Nobel Prize. So initially, the institution was focused on heart disease. That was the first institute. It's still the #1 killer for people in the United States. But over the years, as it's grown, it's really tried to take on some of the most pressing human challenges of the day. And so HIV, infectious disease, was a focus and is a focus. Brain diseases, which you'll hear about tonight, and then finally cancer and inflammation. In addition, a lot of our scientists at Gladstone have really been pioneers in some of the cross-cutting technologies that we need to be able to really solve these problems, including artificial intelligence, CRISPR gene editing, stem cells, and proteomics. We've been affiliated with UCSF, that's where I did my clinical training, ever since the institutes were formed, but we've really formed partnerships all throughout the Bay Area in this ecosystem, including with Stanford, UC Berkeley, the BioHub, the Parker Institute for Cancer Immunotherapy. And if you don't know anything about Gladstone, we are mission-driven. And one of the things that we want to do, and that I'm determined to do before my tombstone is written, is really have an impact for patients. And so part of that is really trying to think very carefully about moving the things out of the laboratory to the clinic to be able to help people. And so with that in mind, we formed a lot of partnerships with pharma, tech, as well as VC companies and creating new companies. Well, I don't know what a bad day at the office looks like for you, but I can tell you what it has looked like for me. And that's when I have had difficult conversations with patients and their families to deliver the news that they have a terrible disease and we have nothing to offer you. And unfortunately, in neurology, there are a number of diseases that sort of fit into that category, including Alzheimer's disease, Parkinson's disease, and ALS. So tonight, I'd like to take you on a journey to tell you a little bit more about what we're trying to do at Gladstone to change this, to have an impact. And in order to do that, I need to first turn you into neuroscientists, so buckle up. So a few fun facts I think about the brain. To me, I fell in love with it during my training at Harvard and Yale. It only weighs 3 pounds. Think about that, 3 pounds of tissue, and yet think about all it does. Like, it's the source of our memories, it's our personality, it's our essence. Think about like the peaks of human achievement. Shakespearean plays, or Einstein's A Theory of Relativity, or even our own Bay Area Alyssa Liu and her gold medal skating performance in the last Olympics, all really emanating from this amazing structure that can do all these things. And so I've never forgot that. Like, who, for example, one of the things, like, who can remember where they were when they first heard the news about 9/11? Yeah, isn't that amazing? Like, I still remember I was on the edge of the bed watching a TV, and it was a very short news clip, but I have not forgotten it. Even though probably every molecule in the neurons that are responsible for that memory have turned over by now. So how does that work? And you're going to carry that memory until the day you die. I think that's amazing. So as I was suggesting, the brain, one of the key features of the brain is it's made up of circuits that help do things like form memories, and the functional unit of these circuits is the neuron. And one of the things that neurons do is they receive signals from other cells at one end, and then they propagate those signals through the cell by electrical activity to release something called neurotransmitters at the other side of the cell to be able to communicate. You have 100 billion neurons that make up your brain, and it can perform 1 billion calculations per second. It's still the most powerful supercomputer in the universe, sort of cubic centimeter for cubic centimeter. And so I think it's understandable that a lot of people call this the most complex machine in the universe. And I think that's one of the reasons why these diseases are so devastating. At Gladstone, we work on all, I think, the major adult-onset neurodegenerative diseases, as well as some forms of mental illness. And I just wanted to walk through a few of these. Huntington's disease, they have some distinct features. It's 100% genetic, and it leads to these uncontrolled motor movements. Maybe some of you remember Woody Guthrie. He suffered from Huntington's disease. Parkinson's disease affects a different set of neurons in the brain, but it's part of the same circuit. It almost produces the mirror image of Huntington's disease. So instead of having too many movements, you have too few movements. And one of the things about Parkinson's disease, it's the neurodegenerative disease on this slide that's increasing the most rapidly of all of them. And we don't really understand why that is. Multiple sclerosis, normally thought as sort of an autoimmune or inflammatory disease, ultimately does lead to neurodegeneration, and it affects the insulation around the neuron that enables it to conduct that electrical activity. It is distinguished by the fact that, on average, it affects people the earliest of all these diseases. ALS progresses the most rapidly, and it affects the motor neurons in your system, so eventually people basically get locked in because they can't move their muscles, their arms and legs. And then Alzheimer's disease, of course, is the most prevalent neurodegenerative disease we have, and probably many in the room have been touched by it indirectly or directly in their family. This disease affects our memories and really destroys our ability to think. So a question I get asked a lot is, what can I do today to be able to reduce my risk of developing these? And the good news is, and this is something we've just learned really in the last decade, is that up to 40 to 45% of dementia is preventable. And so the thing you need to do depends on how old you are. If you're young, you really want to get a good education. That seems to make the biggest difference in terms of reducing your risk. Midlife, basically, follow the advice of your primary care doctor. So diet, exercise, don't smoke, cholesterol, things like that. If you have depression, get it treated. And I think depending on the person, talk therapy or medical therapy or both can be very effective for some people. And then I think later in midlife and in late life, the theme is stay connected to the world you live in. So if your family tells you need a hearing aid, get one. It turns out that your ability to stay connected to the world through hearing and through seeing is really important for the health of your brain. Your brain needs that stimulation to be able to stay engaged and really understand what's going on. And I love that you guys are part of a club. That's one of the reasons why I love coming to talk to this, because social isolation is a huge, as we learned in the pandemic, a huge negative impact on your brain health. And I started a group with a bunch of men, I guess 22 years ago. It's the best thing I ever did. I didn't even think I needed it at the time, but I'm so grateful I did it. It's really been a big thing for me in my life. So what causes neurodegenerative disease? Well, I think origami is a really great way to sort of illustrate one feature, and that is that proteins need to fold properly to function, and if they misfold, they are deleterious. So like origami, where you start out with a plain sheet of paper and it can look the same, you can fold it up to make very different things. So proteins also are a little bit like pearls on a string. They can look like very similar when they're unfolded, but then they need to be able to fold up into specific three-dimensional shapes for them to work. And in neurodegenerative disease, not only do the proteins misfold, but those misfolded proteins cause a loss of the function, oftentimes, of the protein that they're supposed to make, but they also can cause toxic effects on the cells that they're in and lead to neurodegeneration. And so even though the proteins that are affected are different for each of the diseases that I told you about, the common theme is that they misfold. And in fact, in many cases, they are a special form of protein that's prone to misfold. And so why? What causes protein misfolding? Well, one is external stressors. And maybe one of the stressors you've heard about that I'll talk about later is called oxidative stress. Maybe you've heard also of the effects of pesticides. And actually, it's thought that pesticides cause protein misfolding through oxidative stress. Aging turns out to be the number one thing that seems to affect the propensity and risk for these neurodegenerative diseases, and possibly through its effects on the ability of cells to clear these proteins. But genetic variants that we each harbor, depending on the deck of cards we got dealt when we were born, can also affect our risk, either by changing the risk for these proteins to misfold themselves or affecting the cell's ability to clean out the trash, basically. So that's an important mechanism, too. So another thing I want you to leave with tonight is that protein aggregation is a common thread. Well, sometimes it's heartbreaking discussions with patients. They say, you've been at this for years, and what do you have to show for what you've been doing? And I try to assure them it's not for lack of trying. There have been many things that have been put into, especially non-human models of disease, in this case, mouse models of disease, in this case, ALS, but I could have made this slide for Parkinson's or Alzheimer's disease just as easily. All these went on to fail in clinical trial. They looked good in a mouse, but they went on and failed in a human, except for 3. Shown here. One of them actually works pretty well, but it only works in the 2% of ALS patients that have a special gene mutation that this drug treats. The other 2, riluzole and edaravone, probably only add 2 to 3 months to a person's life, so they barely work. So why such a trail of tears? Well, I think one of the challenges in neurodegenerative disease is that there's still some very basic questions that remain unanswered. So when someone sees me and they have ALS, I can't tell necessarily if they have ALS because they got a gene mutation or it's some so-called sporadic disease where we don't know what the cause is. And it turns out 90% of patients with ALS, Alzheimer's, and Parkinson's fit into that category. We really don't know. They look alike clinically, but we don't know what the cause is. And so the key question is, will a drug that works for one of those patients work for all of them? Or are we going to have to know it are there subgroups of patients where we're going to have to tailor drugs to each of those subgroups? That's the nightmare scenario. Because if that's true, the way we do clinical trials right now will never detect it because we enroll everybody and we don't have the statistical power to detect that small subgroup effect. Well, it's not all doom and gloom. So at Gladstone, one of my colleagues, Dr. Shin Yamanaka, developed a new technology called induced pluripotent stem cell technology, which for the first time gives us the ability to take a skin cell or a blood cell from a patient with these sporadic diseases and actually make them into neurons that we can study in the lab to better understand what they do. So in other words, we can model a disease we never could model in a non-human model like mouse, but for the first time in a dish. The way this works is you can take any cell, but most times people take a skin cell called a fibroblast or a blood cell. And what he discovered were 4 factors that you could introduce into these cells to reprogram them so they lost their identity, their original identity, and become a stem cell. And a stem cell is basically a cell that can become any cell in the body. And since then, we've all become good cooks and come up with recipes to make each of those different cell types by adding certain factors to coax them to become that. Another way to think about it, I think that's helpful, is developmentally, a lot of people think about differentiation as balls rolling down a hill. So the iPS or stem cell rolls and becomes defined mature cells. People thought that was a one-way ticket, but what Shinya showed was that by putting these program factors in, you could actually roll the ball up the hill and then turn it and roll it even back. And what I find even more remarkable is we now have the ability to measure these things called methylation marks on your DNA. So I could take a sample of your skin today and tell you with ridiculous accuracy how old you are. If you do that on these cells, you can take a 65-year-old cell, roll it back up to the hill, and it'll look like a newborn cell. So by age, but also by the way it works. So it's been totally rejuvenated. So here's an example of using this technology where, a month or 6 weeks ago, these were skin cells from a donor that then got reprogrammed into iPS cells that then got made into heart cells that are beating in a dish at Gladstone. And you can maybe even make out what looks like a blood vessel pumping blood through it. So it's remarkable. And so you can imagine technologies like this. It's really exciting. Could this become a way to make replacement parts when they wear out in you that we could splice in? And our president here today, Deepak Srivastava, has been working on this and done some really pioneering work about not only doing this, but actually doing it while the tissue's still in you. That looks really interesting. We, of course, I'm a neurologist, so we're more interested in the nerve cells, and we've collected iPS and made iPS lines from patients with all of these diseases so that we could make them into the vulnerable cell types in the lab and really begin to both use them as models to find the causes of these diseases, but then also do screens to be able to identify genes or small molecules that might mitigate disease and could eventually become therapies. Well, as exciting as this is, we needed to figure out ways to really be able to observe that disease process so that we could do some of those experiments. And so when I joined Gladstone a number of years ago, one of the first things I did was invent a technology that we call robotic microscopy to do just that. And so it's got a lot of automation, an automated plate condominium, a robot that transfers plates to and from the nest to a fully automated microscope. But the key thing is we programmed the microscope so that it knew where that plate was in space. So what that meant is you could pass it back and forth and go back to exactly the same position and even the same cell as often as you wanted and for as long as you wanted. So it was like you're doing a clinical trial in a dish, but with neurons instead of patients. And so we use that to basically create movies of these different neurodegenerative diseases to better understand how it all works dynamically. And this is a summary of many years of work where we are looking specifically at this protein misfolding problem. And one of the very surprising things, it completely upended the field, was that the inclusions or these protein deposits that sometimes form can sometimes be a coping mechanism of all things. So sequestering off forms of these proteins that are even more toxic than those deposits. And this has helped us create a blueprint to now think about ways to rationally intervene, and this has led to new discoveries of compounds that boost the protein clearance or trash clearance part of the cell that cross the blood-brain barrier and have shown positive results in our human models of ALS and Huntington's disease. Okay, so you may be saying, well, Steve, you just told us earlier that you had all these compound failures. So so why is it that clinical trials in neurodegenerative disease have a failure rate of about 95%? And that's after spending $1 billion in 12 years. My personal belief, there's a lot of people that have different ideas, but my main idea is that I think human biology is really, really complex. And whenever we model it, we automatically lose some of that complexity. So humans have 20,000 genes as just the minimum part list. It gets even larger if you consider the messenger RNA and the protein isoforms that get produced. And to complicate things further, the relationships between these parts is really complicated. If you look at the pathways, there's divergence, there's convergence, there are feedback loops. This is a diagram of metabolism. Just metabolism. So that's just one pathway, one biological pathway. And I defy you to predict the effect of changing that one molecule right here on the rest of the network. And that's really the therapeutic challenge. You have to target one thing, and you have to predict what it's going to happen, not just even for this pathway, but for the whole organism. Very hard. And then as you neuroscientists learned earlier, we not only have 100 billion neurons, but they make 100 trillion synapses, and those change constantly, like even with experiences you're having tonight. If you didn't sit in that same seat before, one of the first things your hippocampus did as you sat down was have place cells that mapped the entire room without you being aware, so you would know how to get back to that seat if you left it. And so this is going on all the time. And so this is kind of the backdrop that we're working against. And then we bring science. And sometimes I feel like I brought a knife to a gunfight because science is slow, it's expensive, and it relies heavily on simplification, which we call reductionism. And the big challenge is that with with complex systems, reductionism really is a catastrophic mismatch. Basically, you're going to learn insights from simple systems that really will be irrelevant when you try to apply it to more complex systems. I play a little chess. I'm not very good, but I really like to play it. And I'm struck, there are only 32 playing pieces. And yet if you do the calculation, it's 10 to the 120th game boards that are possible. So someone even as smart as Robbie, playing 24 hours a day, 7 days a week, speed chess, it would take more time than the universe has existed to even play all the boards. And this is kind of how we do science. It's sort of one move at a time. We wait to see what we've done, and then we make the next move. And so, honestly, it weighs on me at night, like how much, we don't have much to show, like with Alzheimer's disease after 100 years of work. How much longer is it going to take if we stay this course? Do we need to rethink maybe the way we're doing science? Can we accelerate this? And so, after we built those robots, we built them to unravel cause and effect mechanisms to answer a biological question. But in 2013, I got a call from Google, and they just basically said, "You generate enough data to be interesting. Would you like to be our first academic collaborator?" Sure. So they assigned me 12 engineers, and we began really using AI. And the initial results were just jaw-dropping. We were seeing things in our data we hadn't seen before. I initially then hired a big computer science team, and so now we really have a dedicated effort there. And I'll just tell you, we've now done this for, 13 years, 14 years. It's the first time in my career when I finally felt like I had a tool that was powerful enough, it was commensurate with the complexity of human biology. And so I think this is a golden time in science. I think this is just very, very exciting. So just to make sure we're all on the same page, because you basically, can barely look at any news source without hearing something about AI. That's a term that's been around since the 50s, and it just refers to the ability of a machine to imitate intelligent behavior. Machine learning is a form of AI, and the key idea here is it learns from examples. So instead of having to write computer programs where you instruct the computer to do X, Y, and Z, basically you just show it things you want it to do, things you don't want it to do, and it essentially writes its own code. And it'll do what it is you're trying to teach it. And then deep learning is the thing that's really blown up in the last 8 or 10 years. And that's basically doing this task, but using neural nets. And that's a lot of what I'll talk about for the rest of the talk. So, we care about neurodegeneration, and one of the things that the people in my lab tried to do was look at images and be able to tell whether a neuron was degenerating or not, or about to degenerate. It was awful. They were basically 70 or 80% correct, because we knew we could follow the cells long term. We finally made a biosensor called the Genetically Encoded Death Indicator, or JEDI. It's green when cells are healthy. It's green and red. Oh yeah, JEDI. And they're big Star Wars fans, so in case you were wondering. So it's green and red and looks yellow when cells have made an early commitment to die. It's the earliest thing we found that actually, and it's basically 100% specific. We followed hundreds of thousands of cells. When they light up yellow, they will degenerate. And so we wondered, could we train an AI to be able to tell what humans couldn't from just the morphology of cells? And so we used our robots. We collected tens of thousands of examples. And then showed that to the AI model. I don't know if this will excite you or depress you. 20 years, and humans basically plateaued at around 75%, 80% accuracy. 2.5 hours, and it got to 100%. So now we have an algorithm, and it works ridiculously well, both in vitro and in vivo. And I could just tell you story after story of ways that we've been able to use AI since then to analyze our data, find new genetic cause of diseases. We've been able to show AI cells from patients with different diseases, and it can diagnose disease in a dish. We're hopeful maybe that'll be a diagnostic at some point. We've used it to screen drugs and find new targets with success. So we're super excited about what it can do. And honestly, it just keeps us asking, what else could it do? And one of the things was, well, we've been using it just to analyze data. Is there some way we could basically put it in charge? In other words, could we sort of build a machine a little bit like Google built a chess player that could beat everyone on the planet with reinforcement learning? And I don't think it's such a crazy idea. Biology is complex, but it does have some predictability to it. And like speech, biology and disease have a beginning, a middle, and an end. Could we harness that somehow, particularly nowadays with special AI architectures that underlie large language models, to really be able to understand the language of biology? And so that's the basic premise for what I'll tell you about the thinking microscope. Could we build a machine to learn the language of biology and disease so well we could change how the sentence ends? And so we've got a prototype now at Gladstone called the Thinking Microscope. It's built on the sort of robotic microscope technology I showed you before. We've put in a special piece of hardware called a digital micromirror device. These are thousands of tiny little mirrors about the width of a human hair that we can place under the control of a computer. And when we put that on the light path, we can bounce a beam of light onto the plate, even onto individual cells or even subregions of cells. And as long as those cells contain a molecule that can receive that light and convert it to a biological response, we have a way to do experiments without humans. So it's an autonomous experiment machine. And we've got it so precise now, we can not only hit one neuron, we can hit one neuron independent of its neighbor, or we can selectively hit its neighbor and do a different experiment. So with a system like this, for the first time, we can do thousands of individual experiments in a single well. It's already accelerated our science 100,000, a million-fold in some cases, and so really generated amazing training data sets. So we have a CPU computer that instructs our microscope to collect imaging data and do the experiment. The data gets sent to a GPU computer that uses our AI analysis tools to be able to get insights out of there and begin to generate hypotheses. It then, based on that, starts to design the next round of experiments and send those instructions back to our CPU computer and generates this closed loop where through reinforcement learning, we can get the system to begin to make discoveries and understand biology. Let me show a really concrete example. So we were using this to study a pathway called oxidative stress that I mentioned earlier to you. It's a common thread in neurodegenerative disease, but probably also plays a role in normal aging. And what you're looking at here are videos of cells. This is in the unstimulated state with that JEDI biosensor I mentioned to you. So a lot of healthy green cells, a few red cells. But now what you're looking at in this middle panel or in the right panel, the thinking microscope has gone in, identified each cell in the well, essentially given it a social security number so it keeps track of which cell is which, and then randomizes the cells to treatment or control, a little bit like you would a clinical trial. And the blue is the actual light being used by the thinking microscope to do an experiment on that cell. And you can appreciate then how it's stimulating one cell independent of another, and it's deciding what dose to give, how long to give it, that sort of thing. So it's doing a real experiment. And we asked it if it could discover the relationship between oxidative stress and neurodegeneration, and it generated this graph where you see this very dramatic relationship. The stronger the dose of oxidative stress or light the cell receives, the greater the risk that it'll undergo neurodegeneration. So how do we make it think, though? And here's, we're doing this a couple different ways. I'm just going to tell you one tonight where we used a large language model, ChatGPT, just so that you can see it think out loud. And so the first thing we do is we have the microscope go through and get statistical measures from each of the cells that it sends to ChatGPT, and then ChatGPT starts its process, and it makes its reasoning auditable so we can actually watch it on the screen. So you can see it start to make observations about these cells. It begins to analyze them and generate hypotheses, and then ultimately, based on what it's seen, it starts to design and run next experiments. And then that goes back to the CPU computer, the experiments get executed, it then does a whole nother round, and it's already beginning to see trends here in the data and be able to make observations. So As far as we know, this is the world's first thinking microscope, and even in just the first experiment, we've been able to discover that a small amount of stress can actually make cells more resilient to later stress. It's essentially what doesn't kill you makes you stronger. It's a biological phenomenon called hormesis that took biologists about 20 years to discover, and it did it on the first experiment in a week. So I'm very hopeful that approaches like this can be really useful for both understanding the causes of disease and finding treatments. So I'll finish my talk. I don't think these technologies or what we do necessarily will change the way the sentence starts. So there will be difficult conversations, but I sure hope that these approaches will change the way the sentence ends and that it'll lead us to things that we can give to people that will meaningfully make them better. So I want to end there, and just a huge thanks. Thank you for coming out tonight, just the sort of the support, community support for science. You can meet a real scientist, Lisa Leah, back there from my group. You should say hello to her, and she's brought her son as well. To pull this off, I really want to underscore, I mean, I'm the guy who gets to give the talk, but this is absolutely a team effort, including great scientists like Lisa in my lab. But I also want to say a special thanks to patients and families. We depend entirely on them and their willingness to be able to participate in research, contribute samples, play a role in clinical trials to really see whether any of the things we've done are making progress. And it's a huge sacrifice because in most cases, they won't necessarily benefit themselves from that participation. It's hugely sacrificial. I do think, though, it does take innovative science, and hopefully you got a sense here. I left a tenure-track position to join Gladstone in part because I thought this was the place that could really make a difference. Innovation, they really fostered that, and it was a tremendous community, a lot of smart people, and extremely collaborative. And then finally, I want to say a special thanks to philanthropists because one of the things, science happens to be under attack right now in the United States, and so it's a really difficult time for scientists. On top of that, historically, it already was pretty risk averse, so more incremental changes. What philanthropy has allowed us to do is to do catalytic high risk, high reward work that can really leapfrog ahead, like hopefully some of what I showed you today. That's just not possible with conventional mechanisms for scientific funding. And I want to say in that context, a special shout out to the Koret Foundation, who gave a grant that really helped make this thinking microscope a reality. And so thank you very much for your attention. And I hope you'll come visit us. Robert back there is here from the Gladstone Institutes. I think we give pretty good tours. You can see the thinking microscope and beating heart cells. And I think, for us, it's really important. Tell your friends about Gladstone, help, support science in the way that you can. Come learn more about Gladstone. Maybe you'll get excited and want to get involved in some way. I think it's an amazing place, and I think we all are very passionate about making a difference for people suffering from some of these horrible diseases. So thank you, and that's it.
What a story.
Yeah.
I wanted to say that one of the things that has struck me so strongly about the Gladstone Institutes, and it was confirmed just now, you're not alone. The president, Dr. Deepak Srivastava, is here. Other people from the Gladstone are here. The Gladstone is so good at being able to communicate to the public. And I've seen it happen over and over and over again. So I think that everyone who's in this room who is not already supporting their efforts should step up to the plate, including those of you who are online and including the millions of people who will watch this video around the world. The Gladstone deserves great support because of your communicative capabilities. Oh. So another round of applause, please. Really good.
Thank you. Thank you.
Wow, there's so many questions, and I know I have some on my phone. I'm going to ask a question that is derived from what you said, but I know it's something that people do think a lot about. When you ponder all these dreadful neurodegenerative diseases, what do you think the future of prediction and prevention is? Because so much seems to be geared to therapeutics, diagnostics and therapeutics. Can some of this AI technology help with prediction and prevention?
Well, it certainly can help with prediction, I think. I think that really is what it's especially designed to do, is to be able to take data of one sort or another. It could be a sample of your cells or something else, and to be able to predict what your future might be. We've already been able, in a dish, to predict the future fate of cells using AI, just by connecting an image of what they look like today with images of what cells like that look like in the future. So I do think that that will be possible. I think prevention, it's a great question, and there's a lot of effort and interest around this. It's a complex problem, and I think finding what the thing is that you can do that will actually prevent it will require understanding it, I think, a little bit better.
So do you think the thinking microscope is already stimulating across the spectrum of scientific research, the application of AI? So are people watching what you're doing and thinking, aha, not only can I beat him at his game, but we can apply it in cardiovascular or blah, blah, blah?
I hope so. I do think it's an interesting question because most of the people who come into science come in it from the biology side, and biologists oftentimes don't like math. So computer code is not their natural language, and so I do think there's a bit of a cultural change that's going to occur. But to your question, I literally helped organize a meeting in Boston last week. And I organized a session where I had Anthropic come and introduce them to Claude Science, in part as an effort to democratize some of this technology and to put it in the hands of scientists. And they were so excited. And Anthropic was very nice about giving them an on-ramp and ways to get free compute and things like that. So I'm hopeful that I can show examples like this and get people excited enough to think, yeah, it's worth actually trying to do the work. And the students, the young people are much more ready to do this. A lot of them already know, how to code and things like that. So for them, it's not such a big leap, and they're just, very enthusiastic. Some of the older folks, it's going to take a little bit more persuasion. And yesterday in Princeton, it was exactly that. I think I was the only guy in the whole day talking about neuroscience, and one of only maybe two people talking about biology, but I think a lot of people came and are very interested in applying it for their favorite problem. I think that's been my approach, has been, this is what it can do. Think about what it could do for your problem.
Right. Well, I was listening very carefully to what you said, and I'm not sure how many people noticed this, but you said, well, Google contacted us because we had generated so much data, and they decided they were going to support you. Now think about what that sentence is saying. Most of us over a certain age are used to thinking of medical research with medical doctors, PhDs, hospitals, and we have a tech company called Google who suddenly steps up to the plate and says, we want your data and we want to use it and help. So I'd like to ask a question about this convergence of biology and technology, and tech companies who can't wait to get into the healthcare business or whatever, but historically have not done so. So all of a sudden, Anthropic and ChatGPT are probably knocking on your door. You probably have messages right now on your phone. Okay, so tell us a little bit about that. It's a relatively new phenomenon, isn't it? Non-medical institutions suddenly stepping up to the plate.
Yeah, I mean, in Google's case, it almost sounds quaint, but their first question, their motivation was to see, would AI be helpful for biomedical research? They wanted, are there problems that we could do 10 times faster, or something we could do that humans can't do if we work together? And so that was the sort of origin, and the answer is clearly yes. And so I think they've been excited, and now they've developed the co-scientist. You're seeing some publications in Nature and things like that. And I think increasingly, companies like Google or Anthropic or OpenAI are seeing kind of what I said, that part of the impasse here is the complexity of human biology, and they have the toolbox to really help in many respects. I think, and it was interesting, they had an open, they announced Claude Science, which was a dedicated tool to really help scientists, but they had 3 pharma execs on a panel they were interviewing, and already AI has helped them. they think, improve their productivity 30%.
Wow.
And that isn't even on the biology side. That's
Yeah.
just on, just organizing a clinical trial, analyzing those data are very hard to do. So I think that I'm very excited, about what the future holds and what the impact will be. And so I'm trying to encourage people to really participate. It will take a cultural change in academic science because the way scientists used to do experiments isn't a good match for these AI tools.
Yeah. Barbara, you have a few questions from the audience. Thank you. And I have one online that's been sent in, and here goes. I work on base editors.
Yeah.
Okay. I work on base editors using CRISPR. How can thinking microscopes be useful here?
Yeah, and I'd have to think about that more deeply.
What's a base editor?
Yeah, So I mentioned before this sort of CRISPR system. So there are systems to go in and actually edit individual nucleotides of RNA, DNA, things like that.
Yeah.
And one thing we have done to kind of connect those two is we have put CRISPR under the control of light. And so now we can optically control CRISPR and do genetic manipulations of specific genes in living cells in real time, and then to be able to see their consequences. It's one of the things we're potentially working with Christina and Katie and others at the Gladstone to see if we can harness that for a useful outcome.
So I'm not mentioning names from the audience, but I don't think they're already here anyway. So you will know this is your question when I say it. Thank you so much for your team for this amazing research. That's not a question. That's an introduction. Can you discuss the research using AI in breast cancer at Gladstone?
Yeah, that's a good question. I don't know that we have a specific project. Do you know one, Deepak?
All the immunotherapy efforts,
Okay.
all solid tumors.
Yeah, okay. So what I was going to say was I think normally in terms of imaging, and one of the big challenges with imaging for mammography has been actually to detect lumps, especially in dense breasts, and to really come up with accurate diagnostics. And there's been heartbreaking stories of people who've gotten the wrong diagnosis, either too late or the wrong diagnosis. And AI has been much better than humans at being able to really detect that. But what Deepak mentioned was that one of the institutes at Gladstone is really trying to figure out ways to modify your immune system to help it recognize cancers. So, at this point, cancers can grow because your immune system doesn't recognize them as a problem. And so what we're doing is reprogramming kind of the recognition machinery of some of these immune cells so that now they can recognize these cancer cells and clear them out or deal with some of the circuits so that basically the immune system does a better job. It's called CAR T therapy, and so AI has been really useful in trying to figure out how do we need to engineer them to be able to be effective.
First off, I'm discovering that we're so used to typing on our computers that we don't necessarily have the best penmanship. So I'm doing my best to interpret what is consisting of hieroglyphics. And I'm not an Egyptologist. Okay, here we go. Are there developments, and I apologize to whoever wrote it, I'm not taking the mickey. Are there developments within the world of AI that may accelerate your work? For example, Discovery Loop? That makes sense?
Yeah, wow, somebody's dialed in.
Exactly.
Yeah, so this is a new company that just got formed like a week or two ago. I didn't know about it, but I woke up and one of my friends at Google already texted me about this company. And essentially what they're trying to do is what we're doing. But I think I'm sure more broadly. But I mean, it includes the who's who of Google. Basically, it's the people who developed their architecture and some of the really big cheeses. And so, in fact, my friends already are plotting a way to connect us. so if you're out there, Discovery Loop, give me a call. Yeah, no, I mean, I think one of the big challenges that we still have the large language model I showed you, because I think it's kind of cool to watch the AI think out loud over your data. So it's reasoning over your data. But, to be honest, I don't think language is actually the best approach for images. Because if you think about it, images contain sometimes ineffable information, and if we force it through a text, I'm sure we're gonna impoverish the data that are in there. And the other thing too about, when you bring an LLM in is that you're gonna introduce all the biases that LLM was trained on. And remember, what we really wanna discover are new things no one's ever described before. Yeah. So we wanna really position ourselves that way. So since we're online, and I'll shamelessly, if anyone out there knows JEPA, give me a call. So I think we want to still be open-minded at this stage to explore other architectures, and I'm very sorry for anyone who's lost their job recently at Meta or things like that, but come talk to me. Some of those guys did pioneer different architectures that don't use LLMs that I think might be really useful for this, and really might, both enable the compute, but also help us reason over images in a way we haven't done before. It's a sort of a novel problem.
So if there's a secret agent from Discovery Loop in the audience,
Yeah.
I encourage you afterwards to come forward, take the paper bag off your head and introduce yourself.
Mike Frumkin's looking for you.
Steve's looking for you. Steve's looking for you. I think you've given us a useful clue. Okay, so I'm going to take the side of young people right now, and this is all happening so fast. And there are students getting ready, to go to college from high school. There are students, undergraduates right now, and this is happening so fast on a weekly, daily, hourly basis.
Yeah.
What is the state? What is the possibility of new young scientists finding research positions? Because it sounds like there could be a dramatic lag between need and preparation, and what's your assessment of that? If you were talking to my dear friend Rich Lyons, the chancellor at UC Berkeley, and you said, well, Rich, this is what you should be saying to your students at Berkeley.
Yeah.
What should they be doing?
Yeah.
Breathe deeply.
I know. It reminds me, I gave a talk in no one's going to feel bad for me, but in Tuscany this summer. Yeah, I know, right? And the second question I got was from a student that said, am I going to be out of a job? and it just broke my heart. And even yesterday in Princeton, a lot of the students were like, well, that was the key panel question. It was like, how do you even prepare for this? Right. And it's a great question. And I guess, so I'll offer you my advice, and I do feel deeply for you. I have 3 boys. 2 went into AI and 1 into astrophysics. And I think the thing you want to do, I just encourage you as a kid, really try to find your passion. I know people have given that advice before and it's been received with mixed feelings. But I personally think in science, if you really want a career in science, you got to be prepared for a lot of failure. And you have to have that sort of strength of character that pushes you through to do the next experiment even after you failed the last 10 times. And so you really got to want it. And I think that does not only require drive, but it requires creativity. And at least for me, passion has been a huge source of creativity. It's been like, I really want to solve this problem. What can I do about it? And then the other thing I would just say is learn to think analytically and critically. That will serve you no matter what you do. The skills and the methods will come and go. Don't invest a lot of time learning those, especially at this stage. Learn these really broader principles, and you will be of great value in the future. And you will be successful. People will always have scientific questions.
Well, that's good. That's encouraging.
I don't know if I believe you.
Well, no, I'm not a young person, so I'm just saying that I'm sure that that's something that they would appeal to them.
Okay, good, thanks.
Sorry. Okay, I've got a good question. I think this is a good one.
Okay.
Your AI microscope can test hypotheses and observe living cells without bias. So, has it surprised you yet? Have you caught the microscope trying an experiment or noticing a pattern that a human scientist would never have thought to look for?
Yeah, so I didn't really show you it tonight, but we've developed light-induced ways to make proteins aggregate in cells, and actually been able to recapitulate cascades of disease, including one of the ones Lisa studies, called frontotemporal dementia. And it's discovered multiple things already that were total surprises to us. And I think one of the things that the microscope does is it does crazy experiments. I mean, most humans, that are sane talk themselves out of doing those experiments because they feel like they're going to be a waste of time. But the microscope doesn't have, it's fearless. So it will try crazy things, and it will try a much wider range of ways to do the experiment that a human would do. And that's when you really discover the things you just would never have imagined, almost by definition, because it is a problem in science. We tend to look under the light post for answers, but that's where everyone's looked, and so it's really, I think that unbiased aspect of it is very helpful.
So when I think of the terminology thinking microscope, I think of a little child as a small thinking human. And at a certain point, if you've had little children, they start thinking for themselves. Like, no, I'm not going to eat those peas. No, I want to stay up late. So is it possible in your mind that the thinking microscope can start thinking without concern for you?
If it did that, it would be like every other member of my labs.
Okay.
That would just be join the group, get in line. I think, fortunately, the way closed-loop reinforcement learning, is structured, you do have a reward.
Yeah.
So it is going to work toward that reward by design.
Yeah.
Now, if we hook it up to an LLM, I guess it could. It could think by itself, could go off the rails, and so we will have to be careful about that.
I just wondered what that word, that terminology, thinking.
Yeah.
You think about it, right? I mean,
Yeah.
it's like, okay, it sounds like it's doing what you're asking it to do, but maybe it's doing more than what you're asking it to do. That's what thinking is, right?
Yeah, I guess, we use the term to mean that something that isn't human
Yeah.
is reasoning over our data. So it's
Okay.
getting the information, the inputs it's collecting, and it's trying to make sense of what it's got. We aren't specifying how it should think.
Okay.
We're giving it the goal in some cases, but it's really up to that agent, whether it's a large language model or some of these other architectures, to reason over the data and see things. And again, for the aficionados in the room, there are different forms of machine learning: supervised machine learning, self-supervised machine learning, unsupervised machine learning. We really want to get it. We think that if Google, when they first met me, was like, their theory was that the main limit of science was the human brain. And so they want to take it out of the loop. I'm not quite as cynical, but I think that there's a lot of advantages to being able to have something, think over your data, look at it, put it together in a way that doesn't have a human bias introduced into it, because it's going to get a data-driven solution rather than a kind of preconceived idea of how things work.
It's interesting. A lot of people will say, "Oh, I'm frightened to death of AI because it'll turn into a monster." And the last time I did a program, I got to the corner down here to cross the street,
Yeah.
and a big black SUV driven by a human being almost killed me, and then drove away. And then a Waymo car, driverless, stopped with proper manners and let me cross the street. And I thought, the machine is more reliable than the person. So there we are, right? Okay.
And that was the origin of Waymo, as you probably know. It was in Google X, and they were like, what's the biggest problem with driving? It's humans killing each other. So let's take them out of the loop and see how we do, Yeah.
Yeah, fortunately, we're not running this from the ER at the hospital. Okay, so here's a question that really looks based on what you said and looks more to the future, okay? At the end of the day, people want to see diseases like Alzheimer's, Parkinson's, and ALS eradicated. How does letting AI steer a microscope speed up the process of finding a drug that actually makes it into a patient's hand or blood vessels?
Yeah, so I'll give you two answers. One is that the way it's done conventionally now is we try to figure out what's different about an ALS cell and a healthy cell. And it just gets, so you remember I used the word reductionism? So people kind of keep simplifying it to a single pathway or a single molecule. And I think that's probably fundamentally wrong. I think it's, when we get these diseases, there are a lot of things that go wrong. It's what we would call a systems problem, not just a single, you're not going to get a single bullet, a silver bullet. But that is the mentality. And so the assays get set up to look for silver bullets along one pathway. And then people are surprised when they put them into people and they don't have a big clinical effect. So one thing AI can do is get rid of the silver bullet mentality, and we've done this already, it can come up with the key features, the key changes in cells that actually distinguish ALS from healthy. And it may not just be one thing, it may be multiple things, but it comes up with an AI-defined AI signature of that disease, and we can use that instead of that single endpoint as the assay for drug efficacy. And my bet, I'll bet a beer, that will give us a drug. If we can move that to something that looks healthy, and we can do that with a thinking microscope already, we can use that as the sort of yardstick, that we're going to find hits that have a greater chance of success when they get put into people. They'll move the efficacy needle enough to be able to succeed in clinical trials. So I think that's something that isn't even something that can be done faster. It's something that uniquely can be done with AI. But at the same time, it can do things faster too by doing thousands of experiments simultaneously in the same well with what's called in-well controls. So one of the challenges with AI is it's so sensitive that if you give it a lot of data, it can tell you that this experiment was done by Lisa, this experiment was done by somebody else in the lab. So it can detect small differences in the techniques that scientists in your lab use because it's that sensitive. So with this approach, basically the way we've designed it, it gets rid of that. So it can really just learn on the biology and not get confused by the person and the biologist.
Well, one of the questions is going to tempt me to ask you to do something that I wouldn't do myself.
So great.
Well,
With that introduction.
well, I'll explain what I mean.
It won't get me fired, will it?
No, it might get your salary doubled, though. Oh, I'm sorry. I didn't say that, did I? No, the reason why I said what I said was it's probably impossible for people to realistically predict the future because so often things happen that we would never be. But so this is a future question. So if we fast forward 15 years, I have a hard time with 15 minutes, but if we fast forward 15 years, what is the single boldest prediction you can make about how AI will transform human health and biomedical research? I mean, 15 years is a big. How about under 5?
Well, in some ways, 15 years is easier because
Okay, 15.
I think you're going to knock the ball out of the park. I think that Alzheimer's disease is going to be largely treatable. Yeah. I think that there should be some.
Say that again.
I think Alzheimer's disease is going to be treatable. It's going to be like a diabetes or
Hey, Rise.
high blood pressure.
Hey, Rise, right here.
Well, I got to produce it.
Wow. If you could
Yeah.
pull that off, we would love you.
Well, yeah, already.
Wow.
Like some of the recent clinical trials with things that knock down a protein called tau look really,
Really?
promising. Yeah, yeah, and we got some other things in
This
the
is
pipeline.
being filmed, by the way.
Yeah.
Yeah, great.
For better, for
New.
worse. Yeah.
Fabulous.
Yeah. Yeah. Is that enough, or do you want me being poor?
Well, I mean,
but I think
the audience is
that's
not
the sort
writing.
of approach. I think it's going to have a remarkable effect. It's going to not only produce tangible things that we want, but it's going to change the whole equation in terms of how risky it is to do drug development. You know, if 95% of the clinical trials fail, Big Pharma says it's like Russian roulette. You know, you just roll the dice, and all that work you did preclinically doesn't really matter. But I think with this approach, it's going to speed that up. It's going to lower the risk. It's going to be more likely that things will succeed. The business case has always been there. It's going to lower drug costs. I think there's a bright future.
So, I mean, one of the major implications of what you said is that surely how scientists are trained is going to have to change dramatically. Can you say something about that?
Well, I think there's going to be less and less wet bench work. And by that, for folks who are in the lab, if you ever see a scientist with test tubes and a pipette, that's what wet bench work is. And we can show you that too. Teach you how to pipette if you want. But I think in the future, that's going to be a skill that's going to be less and less something humans do. We already really try to use robots to do all liquid handling because it makes the experiments better and more reproducible. But I think there's going to be less investment in technique and more investment in picking the questions we want to focus on, analyzing and critiquing the data and the results the AI is giving us, and then also just using our imagination about what is the next, because remember, AI trains on things we've already done and we've already said. So it may not necessarily be creative in the sense that we think of creativity, at least that I think of creativity, coming up with something that's really, that would get a patent, non-obvious, and really put things together, not just putting two pieces together that haven't been put together, something truly new. And so I think, and to me, I find that the most fun part of the job. So for me, I'm looking forward to that.
So, there's the old saying that time flies when you're having fun. It is hard to believe that we have now reached the end of the program, and I apologize to those of you whose questions were not read and, in fact, were not even presented. We could go on for a very long time because it's very interesting. I think I have one final question: is what is the most important point you would like this audience in the room and online and people who watch this video in the future to go away with? What can you leave us with that will stimulate conversation, will cause us to think, and also help us understand why we should support the Gladstone even more effectively? And I'm not being paid by them, by the way. Do you know the answer to that question?
Well, I will just say, I do think we're at an unbelievably special time in science where finally we have the tools we've needed forever to really make a dent in some of these horrible diseases. And so it is a really exciting time. So it's especially heartbreaking to me that science is under attack in the United States, and it's difficult even to get grants funded normally based on the quality of the ideas. So I think that would be the thing I want to leave with you. If you want to make a difference like we do, come visit us and get involved, because I think there's an opportunity to make a huge impact and be part of the team and meet other folks who feel as passionately about their diseases as I do about mine.
Wonderful. And thank you.
Of course, yeah.
Kelly Quigley is back here. Kelly, stand up. Kelly was the architect of a lot of this program today. Thank you so much on behalf of your whole team. And if you want to talk to anyone from the Gladstone while they're still here before they run away, the president's here, the lead scientist is here, Kelly's here, and there's probably other Gladstone board members, etc. Don't hesitate to ask questions. I'm going to come on a tour. If I do, I hope to see some of you there. I've never actually been on a tour yet. I want to come on a tour. I know your COO very well, Bob Obana. And I will do everything in my power to support the Gladstone because I'm just so impressed with what you do. And I don't want to get Parkinson's.
All right.
Thank you.
All right.
Thank you so much.